Thank you to SECPHO for the invitation. And in the next few minutes, I will talk about the development of optical sensors for optimization of food processing control. This research is being performed by the Optical Food Process Optimization and Control Research Group, belonging to the Center for Innovation, Research and Transfer Technology at the Universitat Autònoma de Barcelona. The general objective of the research group is the development of potential process analytical technologies to monitor and optimize certain parameters of interest for food process control. Specifically, we have addressed the development of sensor technologies for cheese, yogurt, and meat emulsion manufacturing, as well as more recently for heat treatment of milk. As a general frame to address these developments, we always start with the identification of a relevant process control problem, focusing on two main aspects: quality of the final product and company profits, or more commonly, a combination of both aspects. Once the problem has been identified, two important pieces of information are required. On one side, knowledge on sensor design, I mean photonics, engineering design, etc. And on the other side, knowledge about food composition, instrumental analysis, and even more importantly, the kinetics of those chemical reactions that are taking place or are being affected during the food process under optimization. This multidisciplinary approach is needed for both the development of the specific sensors and the rest of the hardware components, as well as for the development of the prediction algorithms that are required to obtain successfully a new process control technology. With this approach in mind, we definitely bet for the use of simple and affordable optical components to pursue a low-cost process analytical technology tool for inline or online application. A number of different sensors are developed and tested using light sources such as LEDs or halogen lamps in combination with the adequate light delivery or capture components and detectors. Of course, the sensors will need to meet the legal requirements for application in the food industry, especially regarding those materials that will be in direct contact with the food matrix. Combining light sources, fiber optic cables, proofs, and detectors, a number of different optical measurement configurations can be applied for real-time monitoring, for instance, in a pipe. Light reaching the food matrix will be either absorbed, scattered, or transmitted, allowing the three basic measurement configurations that can be observed in the slide. In many cases, a light backscatter configuration using fibers placed at an angle of 100 degrees is more practical for inline monitoring purposes, while in other cases, side scatter or even transmission could be more convenient. An example of a real-time measurement on a pipe would require the installation of an adequate light source and detector. Initially, water would pass through the pipe and the intensity of transmitted light detected by the sensor would be registered. Later, when a liquid food passes through the pipe, the intensity of transmitted light on the matrix would be obtained. With those two intensities, absorbance can be calculated and used in combination with a calibration curve previously established to quantify concentration of a chemical compound of interest. The nature of the liquid food, if it's transparent enough, for instance, would be the main limitation. A quantitative measurement would more likely represent a change in the optical response with time. In many cases, the analysis of the profile by calculation of the first and second derivatives as a function of time allows to identify time-based parameters that are normally correlated with relevant process control parameters. In some cases, it is even possible to extract useful information about the kinetic of the reactions taking place. A good example of that is shown in this figure. We can observe a sigmoidal increase in the light backscatter signal during milk coagulation. The first derivative permits to identify a couple of inflection points in the light backscatter curve. The procedure allows to obtain information from the curve regarding the three main chemical reactions that are taking place typically during milk coagulation. I mean, hydrolysis of kava casing, aggregation of casing micelles, and the gel filming development. Now that we have discussed briefly our main approaches, let's glance through the main sensor technologies that are under development right now. The first one is a unique application that allows real-time estimation of the gel elastic modulus, G', in the cheese bag. The light scatter sensor is installed in the wall of the vat, as can be observed in the pictures. A model has been developed and patented that allows continuous estimation of G', which in turn permits selection of cutting time based on firming. Nowadays, cutting time is selected by the cheese industry based on a prefixed time or following the criteria of the cheesemaker, which is subjective. In the figure, we can observe that G' is estimated with a standard error of prediction smaller than 1.5 pascals, which means objective cutting time selection based on firming. The system has been tested in a French cheese manufacturing company, and a statistically significant improvement of 2.4% in cheese yield has been observed as compared with a prefix cutting time selection. A number of projects have contributed so far to fund this development. For the sensor second development, we are actually using the same light scatter optical sensor that in the previous application. However, a specific prediction algorithm has been developed for prediction of pH during yogurt fermentation. This allows real-time selection of the pH 4.6 in order to stop the fermentation using refrigeration without the need of sampling the gel every 10 or 15 minutes for offline pH measurement. Additionally, several rheological parameters such as G', G2', and the phase angle can be also estimated using specific algorithms. The phase angle, for example, is correlated with the weighing off, which is an important defect in yogurt. This technology has been protected by a European patent application, and a number of projects have already contributed to this research, and new funds have been secured for validation. The third sensor technology is unique in that it allows monitoring of curd moisture change during stirring inside the cheese bag, right after cutting the gel. To that end, the sensor has been designed to collect light from a large area of sample, extracting information about the mixture of curd and whey. But interestingly, this specific design still allows prediction of the cutting time. In other words, just one sensor installed on the wall of the vat is able to monitor coagulation, estimating an optimum cutting time, and predicting the most adequate whey separation endpoint. A number of projects have been funded in the development, and currently we are trying to scale up the technology for industrial validation. The fourth development is a sensor that monitors the degree of emulsion stability at the outlet of a continuous meat emulsifier, and it can be observed in the picture. This technology will allow real-time estimation of cooking losses during sausage manufacturing. Cooking losses are known to exert an economic impact that has been estimated to range between 5 and 4 million euro per year in Spain. Especially cooking loss prediction has been obtained so far in formulas with and without starch. A number of projects have funded this research effort, which currently is initiating industrial validation. The last development is not based on light scatter, but on front phase fluorescence. The ultimate goal is the development of an inline sensor for real-time measurement of thermal damage during the heat treatment of milk. This technology will allow a better control of the heat treatment, as well as inline classification of milk based on real functionality. Note that milk functionality is critical information for a number of important dairy products. To date, as we can see in the table, a total of 11 heat damage markers have been estimated using different combinations of autofluorescent parameters as predictors, with determination coefficients ranging from 0.7 to 0.99. In the figures, the good of fit for prediction of lactulose, furosin, vitamin 2, and casein micelle size can be observed as example. As previously, different projects have supported the research activities so far, and the next step will be to adapt and validate the technology using a small but versatile heat exchanger. Finally, I would like to acknowledge the contribution of the different members of the research team to the development that has been presented. But before finishing, I would like to invite the audience for scientific interaction. Our group is interested in identifying interesting and relevant process control problems where the lack of a specific sensor could contribute to process control improvements. We are interested in participating as consortium members on interesting research projects on those type of applications. Thank you very much for your attention, and I'm ready to take some questions.
Okay, thank you very much, Manuel, for the high level of your research line along these years and the innovations with cheese, yogurt, very, very interesting. We have the first question from Fernando Maya. He says, hello, Manuel, thank you for your presentation. I wanted to ask you how relevant can be, for example, sub-single cycle pulses in the mid-infrared region, 2 to 8 micrometers. Do you think that there is application for these ultrafast laser sources?
Can you repeat again the range of the wavelengths?
In the mid-infrared, 2 to 8 micrometers. Okay,
well, I am focusing currently on all the research we are doing. We are focusing more in the visible, ultraviolet a little bit, and also very, very near infrared. And longer wavelengths, I think it would be really potential. We would be seeing different information, of course. In most of our applications, we are looking for particles and the interaction between particles, and that's why the visible and also the very near infrared is useful. But I guess that other wavelengths are perfectly fine for exploring other options, giving and providing different information for different type of applications.
Okay, thank you. And the second question was if there is application for these ultrafast laser sources.
What type of sources, sorry?
Ultrafast laser.
Oh, okay. Well, I think so. It depends on the type of application that you are going to develop, that you will need a different type of source, light source. For the most applications we are developing, we are using LEDs, and in some cases also halogen lamps. But lasers, well, we will provide very short, very broad, narrow spectra with a high intensity, and that could be very interesting for some specific applications. In our case, we are not using them, but I think that they are potential for applications that might require that.
Okay, Manuel, thank you very much. The time is over. We don't have more questions anyway. Please write down your email in the chat in case someone wants to ask more in detail. And thank you very much, and congratulations.
Thank you.